And our first speaker is Latimer. She is, are you still acting instructor or are you acting assistant professor now? I don't remember.
I am now officially an assistant professor.
Oh, not even acting anymore. Fantastic. Congratulations.
Thank you.
She's an assistant professor in the Department of Laboratory Medicine and Pathology, if I am not mistaken. Yes?
Correct.
And here at the University of Washington, and she's going to talk to us about TDP-43 and tau proteotoxicity. And it's going to be really bad because it's synergistic.
Thank you. All right, let me share my screen here.
The talk is going to be really good, but the toxicity is going to be really bad.
Yes. Let's hope the talk is good. All right, you can see my screen?
Yeah.
Okay, great. Well, I just want to thank you guys for the opportunity to present today. I'm really looking forward to kind of sharing this work and getting some feedback from people. As mentioned, I'm an assistant professor here at the University of Washington. I'm also a neuropathologist in the Division of Neuropathology. And I'm going to talk about a topic that's really captured my attention for the past couple of years, and that's this idea that there's some sort of synergistic relationship between TDP-43 and tau that's exacerbating proteotoxicity in Alzheimer's disease. So I think most of us know that Alzheimer's disease is this terrible public health crisis, and it's the most common dementing illness. And pathologically, it's classically been defined as by these extracellular amyloid plaques, as well as intracellular accumulations of hyperphosphorylated tau. And for a long time, these two pathologies really dominated the field and our focus on trying to identify potential therapeutic targets and understanding what's going on in AD. But more recently, there has been some attention drawn to another pathologic protein, TDP-43, and its potential role in Alzheimer's disease. And so some of you may have heard of TDP-43 in its context with other neurodegenerative diseases, namely amyotrophic lateral sclerosis and frontotemporal dementia, where it was first described as being a major component of the pathology that we see in those diseases. However, probably over the last 10 years, and really it's been accelerated in the last few years, people have been identifying comorbid TDP-43 pathology in Alzheimer's disease. And the data suggests that it's not just some sort of innocent bystander in the pathology, but that it might actually be contributing to the disease process and exacerbating it. So clinically, what has been reported is that individuals who come to autopsy and have Alzheimer's disease pathology as well as comorbid TDP-43 pathology were found to have had worse cognitive impairment during their life and more rapid declines, as well as more severe hippocampal atrophy than individuals who lacked that comorbid TDP-43 pathology. Also, pathologically, there seems to be some interaction with TDP-43 and the pathologic processes of Alzheimer's disease, and most notably with tau pathology. So here in our center, just looking at a subset of cases in the neuropathology core, what we have found is that if you look at the burden of tau pathology in the frontal cortex, that burden is much higher in individuals who have comorbid TDP-43 pathology. And looking at this in a slightly different way, instead of looking at burden of tau in one region, but instead looking at it across multiple brain regions and how extensively the brain is involved by tau pathology. We do that with a Brock stage. We see that at higher Brock stages, you're also more likely to find this TDP-43 co-pathology. And so just through these observational associative studies in human tissue, what we're seeing is what appears to be some kind of interaction between tau and TDP-43, whether it's one protein sort of aggravating the other, or maybe some common upstream aggravator that's causing both pathologies to be more likely to occur together. And that's something that's really hard to address in human tissue because you're just getting this snapshot in time. And so to really start digging into this potential interaction, I had to move to a model system. And so the model system I have chosen to try to answer some of these questions is C. elegans. And I am a neuropathologist. I study human brains for most of my time. And so you might be wondering why a neuropathologist would want to study a worm that doesn't even have a brain. And so certainly I'm well aware I cannot model Alzheimer's disease in C. elegans, but they are a fabulous model for looking at the proteotoxic pathways that are relevant to very complex human diseases. In particular, they have a rapid life cycle. So studying age-related diseases like Alzheimer's disease pathways is really nice in this model system. They also, despite not having a brain, have a very well characterized nervous system and a transparent body. So you can actually see those neurons in a living organism. So in addition to all of these benefits of working in C. elegans, there are already very robust tau and TDP-43 models in C. elegans. And here at the University of Washington, we have a number of experts in C. elegans modeling, and two of them, Drs. Brian Kramer and Nicole Liachko, are my mentors and collaborators, and they have really done a lot in this field of modeling tau and TDP-43 proteinopathy separately. And so what they do is they panneuronally express human isoforms of tau or TDP-43 in a worm and study the effects that that has on various pathways and functions in the worm. But what I was interested in doing was actually putting the two together in one organism to really try to model what I'm seeing in the human brain, which is that the two proteins are happening together in the same brain regions and potentially even in the same neurons. And so what we found in our first attempt at modeling tau and TDP-43 in a single C. elegans is that just like in humans, it's really bad for the worms too. So as I mentioned, they have these strains of TDP only and tau only being panneuronally expressed. And we simply crossed two of those worms to generate this homozygous tau TDP-43 combined proteotoxic model. And to assess the effects that that was having on the worm, we did motility assays, because as these proteins build up, it affects the motor neurons and then the movement of these worms. So you can do a movement assay to get a really good sense of how affected the worm is. And then Western blots from protein extractions to see how much of the proteins are aggregating in the worm. And so the movement assay we did in this case was pretty straightforward. You place a worm on a plate, give it a certain amount of time, and measure how far it moves. And consistent with some of the prior data, if you just have TDP or tau on its own, the phenotype is pretty mild. It doesn't have a very significant effect on the worm's motility. But when you put the two together, it has this very strong effect that really seems like it's more than just adding together two mild phenotypes, that there may be something synergistic happening between tau and TDP-43. And then to look at the amount of protein present, kind of surprisingly, when you look at the tau and TDP-43 combined worm, there is a marked increase in both total and phosphorylated tau and TDP-43 in this strain. And so the TDP-43 alone has very little TDP-43, the tau has very little tau. And so there does appear to be something going on when you have both proteins present. And I'm not showing any of this data, but this seems to be somewhat unique to tau and TDP-43, as we haven't seen quite this extensive synergism with other pathologic proteins. And so it appears that we have generated a really nice and potentially powerful model for looking at some of the effects of combining tau and TDP-43 and modeling what might be happening in human brain. So the next question really became, can we use this model and try to identify ways to mitigate this interaction? Because that could potentially open up a whole new arm of potential therapeutics for Alzheimer's disease. If TDP is really aggravating this process, then maybe that is another way to go as far as trying to treat the disease, at least for the subset of patients that have both pathologies on board. So can this phenotype be modified? In order to go after this, we decided to start with some relatively low-hanging fruit, and that was to use a modifier that has already been very well characterized by the Kramer lab as a modifier of tau pathology. So the Kramer lab identified SUT2 and has published on that as this very potent modifier of tau pathology. When you have SUT2 loss of function introduced into your tau transgenic worms, it protects against tau movement dysfunction, which is shown down here. So here is tau, a tau worm strain that's very much affected, doesn't move as much as the normal worms. And then when you introduce this SUT2 loss of function, it almost completely ameliorates that movement dysfunction. They've also shown that it reduces insoluble tau and ameliorates tau-associated neurodegeneration. There has been less done looking at the effects of SUT2 on TDP-43, but some unpublished data shows some similar, although less potent effects of SUT2 loss of function on TDP-43. So here you can see there is a marked decline in movement in a TDP-43 worm, and then when you have the SUT2 loss of function, it rescues it somewhat, but not nearly to the extent that it does in a tau worm. So it's really unknown whether SUT2 would have a similar effect on the combined tau and TDP-43 model. So to do this, we actually had to do two things. First of all, our tau TDP-43 model that we generated was a very severely affected worm, and that made it very hard to work with. They were not very viable, and so we had to try to dial back the pathology so that we had still a phenotype, but a worm that we could work with. So I'll talk a little bit about what our newer model looks like, but then the main thing that we're focusing on is SUT2 and what loss of function of SUT2 does to this combined model. And we're doing that by looking at the biochemistry, so the total and phosphorylated tau, the effects on the worm's function, particularly its movement, and then also looking at neurodegeneration. So as I mentioned, we had to create a milder tau TDP-43 model. And what we found here is that by having these very low expressing tau TDP-43 worms, when you combine them, we still get this robust increase in tau, both total and phospho-tau, but we don't see that same increase in TDP-43 pathology. So there is TDP-43 pathology on board, not very much phospho-TDP-43, but it does still seem to be enough that it's instigating this increased tau pathology somehow. And then the SUT2 loss of function has some expected effects here, which is in our tau-only strain, it does markedly reduce the amount of total and phosphorylated tau. And also nicely, it does the same thing in the tau TDP-43 combined model. So you get very similar reductions in the tau pathology. So SUT2 remains a very strong modifier of tau pathology even in this combined model. With TDP-43 though, we don't see quite the same effects. And that may be because it's just such a low expressor of TDP-43. In TDP-43 only, we see a mild decrease in our total TDP-43 as a function of the loss of SUT2, but we don't see any effect in the combined, or at least no significant effect in the combined model. And then there's no effect on phosphorylated TDP-43. However, these, as I mentioned, these levels are pretty low. And so I think future work will focus on kind of futzing around with how much tau and TDP-43 is being expressed in the combined model. To look at movement in this series of experiments, we used a slightly different motility assay. This one is a liquid motility. So the worms are placed in liquid, and a normal worm will have this bending motion of their body, and you can count how many times they form this sort of turn, and their turns per minute would give you a sense of how much they're moving, and you can compare that to what a normal worm does. And what we saw here is that although the TDP-43 only worm does have an effect, it has reduced motility compared to a non-transgenic worm, SUT2 loss of function had no effect on that deficit. Consistent with the prior literature though, SUT2 loss of function does nearly completely rescue that tau-only deficit that we see here. And then sort of the new finding is that this very severe tau plus TDP-43 motility deficit is partially rescued by SUT2 loss of function. And so I think there's still a lot of work to do to try to figure out what's going on here, but one thought is, perhaps there is sort of a separate mechanism by which TDP-43 is causing some issues for the worm that SUT2 is just not able to ameliorate. And so most of the rescue that we're seeing here is because of its effect on tau. And then finally, because these worms have motility deficits, we assume they have some negative effects on their motor neurons. And so we can actually visualize their motor neurons to get a sense if that's true or not. And there are strains of worms where their GABAergic motor neurons are tagged with GFP. And so if we cross those with our now triple transgenic worm, we can visualize their motor neurons, which lie along the ventral aspect of the worm, and count them to see if we've lost any neurons. And so we did this blinded to their genotype on day 1 and day 4 of adulthood, and what we saw consistent with prior literature, there's a loss of neurons in the tau-only model that is much more severe when you combine it with TDP-43. And then SUT2 rescues the neurodegeneration nearly completely in the tau model and to a slightly lesser extent in the tau TDP-43 model. And so just some general conclusions and future directions for this work. It's really still pretty preliminary. We're still sort of kind of futzing around with the combined model. But in general, it does seem like C. elegans is a very strong model system to be able to study this combined proteotoxicity, as well as investigate potential mechanisms and modifiers. The data that we have in hand suggests that the SUT2 loss of function is rescuing tau and TDP-43 deficits largely through its effects on tau pathology. And that what this more mild phenotype is showing us is that TDP may actually be the instigator here, and it's increasing tau aggregation and phosphorylation. And so future work will be to return to humans, assessing SUT2 immunoreactivity in human brain to look at the differential expression in individuals with different degrees of TDP and tau pathology, to look at additional known modifiers of tau or TDP-43 on the combined model. And I'm particularly interested in looking at strong modifiers of TDP because it would be interesting to know if that has a stronger effect if TDP truly is the instigator here. And then identified novel potential modifiers of tau and TDP through transcriptomics work in both humans and worms. And so with that, I don't even think this covers everybody, but predominantly the Neuropath core, the Department of Lab Medicine Pathology, and my collaborators at the VA, as well as the ADRC, the ACT study, and my funding sources. And so with that, I will conclude, and I'm happy to take questions or have some discussion.
Thank you so much. It was a great talk, and it wouldn't be a Geroscience symposium without the talk centered on C. elegans. We have a number of questions in a few minutes. We're going to try and address as many as we can. On the Slack channel, Anthony Grillo is asking, is the TDP-43 and tau co-pathology in Alzheimer patients normally due to an underlying disturbance in a single upstream kinase common to both, example TTBK, that can be targeted, or is it thought to be primarily caused by two independent pathways? If so, do you think silencing TTBK in the tau and TDP worms would prevent the synergistic effects you described?
So that is an excellent question, and it's one of the questions I think we have. We don't know. This is really, even though we've been describing TDP-43 in Alzheimer's brains, the pathology community has for 10 years, it's really only recently been appreciated, I think, and the people are really looking into it. So I think that's one of the top questions, and actually my K08 is focused on trying to look at a number of these known modifiers, and the kinases are on my short list of genes to look at, because I do wonder, is it something upstream that's setting you up for both, or is it one sort of pushing the other? So that's definitely on the list of things to look at.
All right, and then we have Adrian Wang. Hey, Adrian. Great talk, thanks for sharing. Maybe I missed this, but can you speak to how SUT2 knockout might be ameliorating tau pathology? Do you think your data supports an actual interaction between tau and TDP-43, or might these just be two parallel ways to paralyze and gum up proteostasis in a worm?
So I guess to the SUT2, I don't know if we know that, and Nikki is presenting next, and she might be able to comment more on that. My understanding is we weren't quite sure how it was ameliorating it. But as far as whether the data suggests that there is synergism or separate pathways, I don't know that it strongly supports either at this point. I do think that it may be a combination. It does look like that having TDP on board is pushing tau. And so to me, that's kind of a synergism there, that if you have TDP, you're somehow exacerbating tau's pathologies, but that TDP may have an additional separate pathway. So maybe that's a cop-out, but it is likely a combination of effects.
All right, let's do one more. Srimathy Logan, have you looked at soluble and insoluble forms of tau with TDP-43? Does C. elegans model both forms of tau? Does TDP-43 increase ratio of the soluble, hence more toxic forms compared to tau aggregates?
So I am not sure. I know we have primarily focused on the insoluble forms, and then so I don't recall if we have the data on the soluble forms. And I do invite Nikki to chime in if I'm misremembering or forgetting something, but that would be another good area to look at as well.
All right, fantastic. It is 10:30, so we need to go on with the program. Our next speaker is Nicole Liashko. Caitlin, I will direct you to the question and answer tab on Zoom, and there's also one extra question on the Slack for you. Thank you so much.
Thank you, guys.